Thank you, Kevin. Yeah, so just to reiterate, I'm an engineer by qualification. I'm still a professional student, but I am doing a talk today on biology. So if there's someone out there with greater experience, please, if you think you can input anything, feel free. All right. So I know the topic of my talk says DNA replication. I've actually expanded it a little bit beyond that to a gene expression and a couple of extra bits and pieces as well. So just a quick overview. The structure of my talk is going to basically start with what is DNA, the structure and the makeup of it, and where it's situated in the cells, or wherever that may be. So just to give an idea on that sort of thing. Key roles that DNA actually plays within the body and cells. And just going to give a brief idea or discussion on some of the origin, the theory of how DNA actually came to be. It seems like quite a wonderful structure in my eyes, being an engineer, and so yeah, I'll give some ideas on what people have suggested there too. Okay, so starting off, what is DNA? So Kevin put it forward to me to spell it out to you. I won't spell it, but it's deoxyribonucleic acid or something along those lines. It's close enough. Not a bad effort. I haven't actually got it written down somewhere, so I did that off the fly. I've got to hit twice, so I apologise if my computer is ahead of my slides or vice versa. It's a double helix, as you can see over in your far right. Basically we go from an extremely zoomed in portion over in the right, and we get more and more wound and zooming further out as we go right up there towards the chromosome, which is the X, that guy. Starting from the far right, the double helix shape is basically where the information is stored, and we'll cover that a bit later. But the information is stored by chemicals. They're nucleotide pairs, and they form basically the rungs of the ladder. So if you can imagine a digital code or something like that, each rung of the ladder effectively codes for some sort of a process that goes on. Just some interesting facts if you're an engineer, that the full rotation about the axes of this double helix is about every 10 to 11 nucleotides, so it's fair spinning. Not literally spinning, but you know what I mean. And it's effectively bundled up onto spools. These spools are called histones, and they effectively package this very precious DNA. I mean, you don't want too much going wrong with it. These spools are then also bundled up into more packages called nucleosomes, and they are actually coiled. Not coiled only once, but coiled again into supercoils. So you've got a continuous coiling of this DNA information. And it's extremely long. I mean, you might be reading ahead of me here, but so the DNA in one cell is about the height of you, about 2 meters. And you've got a lot of cells, so that makes 177 billion kilometers in your whole body. Some people probably a little more than others, but that's just a fact. And only about 1 to 3% of the actual DNA in our body goes towards making your genes. The rest is either redundant, not used, or it goes towards some sort of other process, which is usually to help with replicating or making your genes into proteins, basically. Okay, so without mumbling, I've already briefly covered the supercoils and coils, but there's a very specific reason for this. For one, it's a good idea to package it nicely, and it's a way to package it and keep it safe. And the second reason is because effectively when you want to read what is actually contained within the rungs, you have to split the DNA apart using some very particular enzymes and then read it. And in the process of splitting it apart, you have to unravel it, and that would actually cause a lot of torsion on the rungs. And so by pre-twisting it, you prevent the DNA from getting damaged. And chromosomes, I briefed that already, but they are basically lengths of DNA. We have 46 of them in each one of our cells, and so basically all the information that makes you you is on one of these 46 chromosomes, and there's actually a redundancy because they're in pairs. They're typically viewable under a microscope, so they're actually quite large in the grand scheme of things. They're still within the nucleus, but when they're dividing, you can see them reasonably easily.
Sorry, when you said DNA defines what's you, it'd be the combination of all 23?
Correct.
23 different chromosomes.
Yes, yeah, so your eye colour will be on one of those 23 chromosomes. Or could be on, it could be a host of things actually. It could be a little bit on all of them. I don't know how many genes go towards your eye colour exactly, but yeah, if that makes sense. So, and there was one last point on the chromosomes. They, or two actually. The centre point is actually used to pull the chromosomes apart during DNA replication. So when the whole length of DNA has been copied, it actually grabs it in the middle using little fibres and actually rips it apart to produce a second cell, so that you've got two identical cells.
Now, excuse me, you're talking about this enzyme ripping it apart, or is it?
They are called.
You're doing it, or is it doing it itself, or what?
They're called spindles. 1 second, sorry. Let me just double check that. Spindle fibres, yeah. I'm not sure actually what spindle fibres are made up of, but they're within the nucleus. So you've got your nucleus, you've got your cell, and then you've got your nucleus, which has got your DNA in it. I assume it's some kind of a enzyme, but I'm not entirely sure on what actually makes up these little fibres, but these fibres pull. Someone may be able to, does anyone know much about spindle fibres? It'll be I'd almost guarantee it's going to be a type of enzyme, because that's what proteins basically are. They're all the building blocks of cells and all that sort of thing. So
you're talking very softly. Can you speak up a bit
sorry.
louder?
Yeah, all right.
Others
I should.
Can hear you properly there.
Sorry. I got a bit of something in my throat, so it doesn't help. The other thing was at the end of the chromosomes, there's actually some something that gets a bit shorter every time, and it's called telomeres. So these telomeres, basically they determine the life of the cell. So every single time you take the cell and divide it, these telomeres get a bit shorter. I don't actually know what the mechanism is behind that, but something like a cancer cell produces its own enzyme, and that enzyme prevents that from happening. So that's why a cancer cell will just divide and divide and divide and divide, because it basically takes this indicator of how old the cell is and it ignores it, and it'll just make it divide and divide over and over. So that's just an extra little interesting point in there, which I never knew about. So
telomere is kind of related to why we age.
Someone said yes. So yes, I would say so. I don't know much about telomeres, to be honest.
Yeah, you said that they decay or get smaller.
Yeah. So
and you say that so when they get copied and produce another cell, then that cell, next cell, slightly degraded compared to the first one.
not sure. I don't actually know how that relates to the age of somebody. That much I didn't look into. That was just it was a bit off a side topic that I just thought I'd throw in there as interest. Yeah. So I was talking about the base pairs. So DNA and RNA, which is related to DNA, they have a similar structure. And so DNA has got adenine, cytosine, guanine, and thymine. So they are the 4 pairs. So like I said previously, I said A will always match to T, although that's what it says up the top here. A will always match to T, and G will always match to C. So that's the pairs. So you'll never have A matching with G or A matching with C. That doesn't happen unless, I don't know, someone knows something that I don't about mutations probably. So that's how it works. And so if you've got, if you know you've got T on one side, then you know that A is going to match the other side, which is a bit of a hint later on for how things work. So that's the actual structure of the double helix. I think everyone can see that. Can you guys see that properly? So a close up on the double helix, you can see the G always matches with the C and the A always matches with the T. Now, just...
Is that hydrogen bonds all the way down?
E for those, yes. In the middle, yes. I'm not sure what the bonds are on the outside, but it's phosphate, sugar phosphates. But of interest is this here's 3' end, and this says 5' end. Now that's got to do with the orientation of the sugar and the phosphate. So these are all sort of facing up, and these ones here are all facing down. Now that is important later on, because one side, they're basically the two edges, they're facing different ways. So, okay, so continuing on, what is DNA within cells? So where's it placed? So just to give you an idea, this is a animal cell. This here is a plant cell. And this little guy here is a prokaryote cell. Now that will become apparent in a second. So we are eukaryotes. Our cells have a nucleus. That's basically all that means. So eukaryote. So we got a. This is us. We've got a eukaryote. We've got a nucleus there. Plants also have a nucleus, which is there. So and prokaryotes don't have nucleuses. They just have their DNA swimming around in their guts, basically. So that's the DNA just sitting inside the cell. There's a summary of the differences. Some of the key points, like I said, the no nucleus for the prokaryotes, whereas we have nucleus. And the prokaryotes have a circular DNA, so it's just a ring, which is interesting. Whereas eukaryotes have got the chromosomes double helix shape. Introns are non-coding genes. So where it says these guys don't have introns, that means that they don't have DNA which is not sort of turned into something, or if that makes sense. So introns are basically taken out of the equation when you go to make something from your DNA. Right. All right, that's enough of that one. Yeah, and eukaryotes are basically just your bacteria and stuff like that, as you could probably guess from the image. Okay, so another interesting point, which ties into origins of sort of the theories of how things came to be, is mitochondria. So this is a eukaryotic cell, and the mitochondria within there is effectively like your power plant, powerhouse. So that's where all the energy is turned into ATP or whatever it is. Now that goes to provide energy for the rest of the cell. Now these have their own DNA, which is interesting. So rather than sort of being a part of the cell and having the DNA stored in the nucleus, they're sort of their own freelance organ, which is interesting to note. And the same thing occurs within the chloroplast of a plant cell. They also have their own DNA. And so they divide and they do all their stuff independent from the rest of the cell.
So you mentioned that there are 46 chromosomes and
Yes.
23 pairs. So how does that kind of relate to the mitochondria?
So the mitochondria has rings of DNA, and these rings of DNA are not included in those 23 pairs.
All right.
So that's completely separate.
Chromosomes at all?
Well, no, it's just DNA. It's rings of DNA. I don't know if they would consider it a chromosome per se.
That is the same type of DNA that's in the prokaryotic cell, bacteria.
Yes.
They have the chromosome, and the idea is that basically our complex cells that we have co-opted,
Sure.
or have developed by co-opting
Basically
these
stolen.
prokaryotic bacteria-like organisms within them that
Yes. Yeah, and
have evolved from
that's
that.
the point I was going to make later.
sorry,
But
sorry.
No, I told you to do that, and I'm glad that you did, because
And it's the same
I
with
was
the plants.
considering whether I bring that up now or whether I was going to jump off topic. But yes.
So it's the same with the plants. You've got a very similar structure
Yes.
in a chloroplast to for example, the plasmodium that causes malaria,
Yes.
which is just a prokaryotic single cell organism,
Yep.
and remarkably similar in terms of the DNA that you get in the chloroplast, which again in plant cells are these independent
Yeah.
little creatures, if you like,
Yes. Yeah.
that
So
operate within the cell.
it seems almost like they were imprisoned, doesn't it? And sort of enslaved, I suppose you could say. And so it's interesting. It's interesting. So that's So that's the finish of the overview of DNA. So just to give a visual representation of DNA replication, this is as simple as you can get when it comes to a diagram for DNA replication. There are so many more things that go on in the back end of this and the front end of this, but this gives a pretty good idea on what goes on. So here we've got our friend the chromosome again, and that is actually unwound by various enzymes. Actually, there's more enzymes at the front end, but the main one is DNA helicase here. He actually rips the ladder apart. So you've got these pairs of these base pairs which are joined with these bonds, and this enzyme goes through and cuts it right down the guts, and so that is then held apart by various other enzymes, but in particular there's these guys are the same thing, DNA polymerase, and they will go in right behind DNA helicase, which is the one the scissors, and match pair to pair. So if there was an A, it would match a T. If there was a T, it would match an A, etc., etc. And so what you effectively get is you have one lot of the information here, but with redundancy, because the pairs always make the same pair. You split it so you've got the same thing but in 2 strands, and then you complete the 2 separate strands to make 2 lots of DNA, if that makes sense. Without getting too silly about things, this leading strand has the best time because he doesn't have to reverse back on himself. Now, the reason I say that is because before I was telling you about how the 2 sides of the DNA face different directions. If it's not facing the direction you want to, you have to do a little bit and then move. Do a little bit and then move backwards. Do a little bit and move backwards. So it literally has to jump, well, it has to run ahead and then come back. And then run ahead and then come back. And then run ahead and come back. So it's got its work cut out, this bottom guy. Same molecule, but because the DNA strands are facing different ways, it has to run ahead and then come back and meet up to where it finished.
What drives the movement? What drives the
bonds, basically. These are enzymes. So enzymes have active working regions. They've got different. Yeah, it's a good. There's not a 4-stroke motor in that thing. It's a good question. Probably.
Is it just a single molecule?
That one, I believe, is actually a DNA polymerase. I think it's that one that is actually, no, I believe that's just an enzyme, yes.
It's a protein molecule.
There are other ones that are mixtures of RNA and enzyme. So an RNA and an enzyme mix together and twist and tangle into some complicated working machine. Yeah. So, and that's ribosomes, which do the next system that I'm going to go over. But I believe DNA polymerase is just a standard enzyme. An enzyme and protein. Enzymes are made out of proteins.
Is it possible that that's curling up, you were saying initially,
yes,
like a big spring?
Yes.
I wonder if that has anything to do with when it's that little gadget breaks the pods.
Sure. Yeah.
Then there's energy within that spring that motivates.
Could be. I'm not sure. I would imagine it would take energy to do it. So there would be ATP or something like that. That would also help. But in terms of bringing the polypeptides in, they float in the goo, and that is not ordered how they come in. So they wait for one to come and make a bond, and then it'll move on. Wait for it to make the bond. So this front end process is random, and then obviously this is very structured after that. If that makes sense.
It's got big squares to make a flower around.
That's right. That's exactly right. Okay. So now that we've seen the general overview, I'll just go through the key points again. So the whole point of it is to make one DNA strand into 2, so that we can make 2 cells out of 1. Okay, so the enzymes that are involved, we've got the scissors, which is the helicase. We've also got the polymerase, which plants the flowers in Kevin's analogy, so that actually brings them together. So that one there is the polymerase, and that's the polymerase there too. So they cause the chemical reaction to happen. That guy also proofreads it and fixes it if something goes wrong. We've also got a DNA clamp. Now, the DNA clamp stops the polymerase, which is the one that I was just talking about, from delocating. So it stops it from actually getting knocked off the actual torn apart DNA. Single strand binding protein, that's a bit of a mouthful, that maintains the strand separation. So if you pull the strand apart, it's going to want to rejoin because it's got bonds there that want to rejoin. This actually holds them apart while DNA polymerase goes through and does what he wants to do. Yeah, I don't even know how to say that one, but he goes ahead of everything and he unwinds it. That original, I said how it's originally sprung up like it is, he goes and yeah, he relaxes the coil. Yeah, and yeah, we won't go into too much more on there. Yeah, occurrence is just the processing occurs along both sides of the DNA, so you already know that. And the product that we get is a duplicated DNA, and this takes approximately 2 hours to do the entire DNA of 3 billion base, not 3 billion total, so 3 billion divided by 23, I guess. Yeah. So it zips through it pretty quick. And post-processing is, yeah, so the only, with DNA replication, because we've got the two strands, we've got one facing one way, one facing the other. I said that one of them had to run ahead and then walk backwards, run ahead, walk backwards. That actually produces a bunch of fragments, and there's another enzyme that goes through and actually stitches them all together. So that's DNA ligase, and they're called Okazaki fragments because it was a Japanese man that actually discovered that. Yeah.
Where do they cover, like these enzymes that actually do the work, where do they come from? Who
DNA.
Sorry?
They come from DNA. It's all coded within your DNA to make things that can reproduce your DNA.
Right, so the DNA produces the enzymes and the enzymes produce the
Yes,
DNA.
correct. That's why it's pretty hard to work out how it works, isn't it? Yeah. Okay, so that's DNA replication. So we can do, that's a very, very basic overview on how a cell can take its DNA and produce 2 DNA from it, right? Now, what do we actually do with DNA? As I've just sort of pointed out, we take the data on the DNA and we use it to make enzymes. We also use it to make RNA molecules. Some RNA molecules can act as enzymes or join with enzymes in a noodle soup to do something else. Now, these enzymes do chemical reactions. Like I've shown you, they take DNA and they rip it apart. That's a reaction, you know, it's making a process happen.
This happens automatically in our body. You're not talking about taking it out and you doing it. You're talking about what our body does with it.
Yeah, while you're sleeping. Yeah, yeah, and it's happening very fast and very often. Yeah. So what the so I'm going to talk about RNA and protein synthesis. So that happens. The first part of that is very similar to the DNA replication. It does the same thing. It rips it apart in the same way, using actually the same helicase does the same thing at the front. So it actually uses the same protein to rip it apart. Now, rather than both strands being used, now we've only got the one ideal strand being used, so rather than getting 2 strands out, we now only get the 1 strand out. That 1 strand codes for a particular gene, a particular molecule, and I'll explain that all in the next slide. But effectively, that's the same process. going on there. We're just not connecting up the other sides. Now, if we only have the half of the helix like this, so it's only that's called RNA. So rather than having the double helix, it's just a single-sided helix. RNA are very interesting. Yeah, so move on there. Hold on, what did I do? I did something wrong here. Okay, so the first part produced an RNA strand, which is just a mirror image of a length of your DNA. That thread of RNA passes out through a hole in the nucleus. So there's a bunch of ports in the nucleus, and it passes through that. I would imagine there's some sophistication going on there, but I haven't looked into that. So that comes out into the main part of the cell, and it encounters what's called, a ribosome. A ribosome is what I was talking about before. It's a protein, but it's also made up of RNA as well in some noodle mash, and it does a lot of really interesting stuff. But effectively, it acts as a workshop, I guess you could say. It allows all these chemical reactions. It holds all these chemicals in place so that it can do what I'll explain to you in a minute. So each lot of 3 base pairs codes for a particular amino acid. Amino acids are these guys up here, and a chain of amino acids is a protein. So if you have 100 amino acids in a row, all linked together, that's a protein. So I couldn't tell you how many amino acids this ribosome has, but it probably has a lot. Okay. So what this the process that goes on, this ribosome pushes them through 3 base pairs at a time, and each time another 3 base pairs comes along, a very specific amino acid to that lot of 3 bases is attached, and it gets a bond to the last one, and then this will move across. So it'll move across by 3. It'll move through the zip 3 spaces, and then it'll find another amino acid that is represented by those 3 bases. And so it'll continually do that until it reaches the end, and the end has got a specific ID as well. That's there's one of these, 3 of these will actually represent an end as well. So it'll tell the ribosome that's it. So that's how a protein is made. Now these strange looking things here are called tRNA. That's actually an RNA. So it's the same as this RNA. It was just produced straight from the DNA, and it did its own thing, and it folded itself, and it became an enzyme. So that's why I was saying RNA is quite fascinating, because not only does it hold information like this does, it actually can fold itself, and it's got the right sort of active areas where it can actually cause chemical reactions to happen as well. If that makes sense. Was anything lost?
Well, just to comment that the way it folds is actually just a property, purely a property of the amino acids and the sequence of them.
Oh yeah, sure.
So, and I don't know if you're going to go on to explain, there's a limited number, there's all those different combinations. You can have a certain number of combinations of 3 letters.
Yeah.
There's a limited number of different kinds of amino acids.
Yeah, there's 20. Yeah,
there
20.
is some redundancy.
Yes.
But with your, and I've forgotten what the number is it 22 or whatever it is?
Yeah, I double checked it because Kevin asked me as well. Yeah. There's 20 amino acids.
But basically, the order you put those 20 building blocks produces a 3-dimensional
particular shape. Yes.
Protein.
Yes.
So it's basically a long chain and you can get really complex structures from it because it wraps around itself. It's actually determined by the precise detail of that order.
Yes.
So that's
correct.
what the code of DNA is actually describing.
Doing.
It's the order
Yes.
of these 22 building blocks
Yeah.
that attract each other and therefore cause this protein to fold in particular
Complex
ways.
shapes,
And it's this shape, these complex shapes,
Yeah.
and the detail of them that actually determine the property and how these enzymes work by reacting, and their shape and their charges of the different amino acids
For sure.
gives the structure
For sure.
to how it all works. So
Yeah.
it's actually chemicals. It's all done with chemicals and their properties and this consequence of the sequences.
Yeah. Yeah. Thank you. That's. I didn't describe it that elaborately, so thank you. Yeah. Did anyone not understand that and wants that reiterated? But based on like the order, you get all sorts of different shapes, and they can do a lot within your body. Yeah.
And they can track different molecules within the cell and bring them together at the right angle to cause this chemical reaction in the cell to happen.
Yes.
And of course, it's all controlled by the timing of when on the DNA, when they're produced, and when they bring those chemicals together to make that reaction happen in the cell. So.
And that's both proteins, and that's also RNA as well. So RNA can simulate that sort of thing as well. It can do that same sort of thing.
Can I just ask a couple, just before we move on? So the RNA came from the DNA splitting, and then that came out from the...
Yes, that's right, yeah. So the RNA came out from taking a direct copy from one of the rungs.
Right, yeah.
And then that passed out of the nucleus into the main body of the cell to then be turned into something. Right. That something being an enzyme or...
Where did the ribosome come from?
That is an enzyme, and it's also RNA. So
okay. So it was a previous...
I believe, I'll just go forwards. I think that is a diagram of it coding there. I think that was a ribosome, maybe. Not sure. But that's effectively what you'd have. So if the yellow was, say, protein, like amino acids, and say the blue was RNA, they have chemically reacted in a way and twisted in a way that they have produced this elegant structure.
In your slide on DNA replication, you have one DNA producing two DNAs.
Yes.
So when is the RNA produced? Like, do they get split again to produce that RNA?
When the cell is going to divide, it has to replicate all of its DNA.
Replicates the DNA?
Yes. So RNA is produced on a different part of the DNA at a different time, I guess. I believe that multiple lengths of DNA can be worked on at the same time. I couldn't necessarily confirm that on my own steam. I'm not sure what your question... I understand what your question is, but it's a different process. You don't make RNA and replicate the DNA at the same time, at the same place.
Does the DNA have to be split again to produce that RNA? Or
Yes, resplit. So after the DNA has replicated itself, it has zipped itself back up.
Yeah.
And so it would then be packaged and pulled to the various cells.
What's the function of the messenger RNA and the transfer RNA that we had in the previous?
Yes. So I think I did brief over that a bit too fast because I didn't want to overload too much information. So the transfer RNA is just an RNA that has got, if I could, it's got the ability to code 3 peptide bases on the bottom. So the bottom part of it looks like DNA, the bottom part of that RNA, because that's what this whole thing is, right? This whole thing is actually these bases, because that is an RNA molecule. It's the same as this. But all of these will be internally bonding to each other, so they're not actually sticking out in the open, like ready to chemically react with something. So these rungs of the ladder, per se, these are all ready to react with something, because they're just out in the open. Whereas this guy, all of these are interlocking. They've actually folded in on themselves, and they've actually it's bonded with itself. But down the bottom here, it actually flips around to display three nucleotides, and those 3 nucleotides match perfectly with 3 along an RNA. So if this one here came in because it smelt that one and it smelt good, all 3 lined up perfectly with all 3, then the amino acid that this guy's associated with would be the next one in the chain.
The particular one that it's associated with.
Yeah.
So it can only...
Each tRNA is built to deliver a specific amino acid, because it has to be, because it's based on the bottom. This is its ID, basically. It's got an identification of 3 base pairs on the bottom, and so if it's got these 3 base pairs, then it's always going to try and match with AGU. And every time it sees AGU come along, it will try and match with AGU and deliver its payload, which is the amino acid. A particular amino acid. So it's
so
a way.
The sequence of the nucleotides determines the amino acid that's going to go in the next.
Yes,
in the leg.
correct. Yeah, that's right. So every 3 nucleotides will give you 1 amino acid. And so then you build up a huge chain of these amino acids, and then that'll spin and contort and do all sorts of stuff and become a sophisticated enzyme, which will do something like the rest of these enzymes do. I mean, this is an enzyme, and it does this whole machine. That's an enzyme. So it's building itself in a way, if that makes sense. Okay. Yeah. So this is just a little bit of extra information. So if you were interested in what the actual amino acid was that 3 base pairs makes, so like you could take your first one being G, and then you could say the next one is C, and then the next one is U. You would say it's ALA, which I don't know what it stands for. I know GLY, I believe, is glycerine. I recognised another one a little while ago. I don't know that many of them, right? They're amino acids. So all of these, so there should be 20 of them. There's more than 20. Well, some of them are doubled up here. And there's also, is there a stop? Or have they ignored the stop? One of these might be coded for a stop as well. Stop and start. I know it's definitely on some charts. There's stops and starts, so I'm not sure which one that represents on this. No, what was I going to say? Yeah, so some of them, like for instance, GC, then you can have anything following that, and you'll always get the same amino acid. So there's redundancy, like 20 amino acids, and there's Thumb combinations, 4 times 4 times 4, something like that. Okay. Yeah, so I've already touched on this, but non-coding DNA, so DNA that doesn't necessarily go to producing a protein, so it doesn't go through this system here, it will still get the RNA made from it, and often that will go into producing RNA. And as I said before, RNA is sort of a hybrid. It holds information, and it also does its own sort of enzyme work as well. So it's quite a clever molecule, quite a clever system. And these are just, I mean, I'll go over them quickly because I think we're running out of time. There are some of the processes that they do, right? So they can be involved in translation. So the translation, which was making the amino acids, RNA splicing, so they can bring things together, DNA replication, gene regulation. So if your body's got enough of a particular something in it, or the cell, it says we don't need any more of that, it will actually, often they will go and they'll actually block that from being done. So they'll actually get in the way of all these things from being produced. Yeah. Okay, just now up to the part where no one seems to have made up their mind on how things work. How did all this come to be? It's all very amazing, right? I mean, my knowledge on the whole thing is very limited. I'm very fascinated with how these things work, and coming from an engineering background, I see all of these machines, and I know they're chemicals, but they're also very much like machines as well. I find it fascinating. Now, there's the intelligent design, or the sort of the God did it, which some people say is nonsense, some people say is logical, some people say it's disproven, some people say it's not. Who do you believe? So there's spontaneous creation, there's also a form of guided evolution. So God sort of clicked his fingers at the start and sort of got things flowing, and then kicked back and had a mochaccino or something.
What do you mean by spontaneous creation? What does that mean?
Oh, in terms of 6 day, and he had a rest on the 7th day type thing. He sort of
done really quickly after short.
Yeah, that's right. Yeah.
Actually, being a Christian and knowing what the Bible says, and knowing that you've just explained to us the complexity of the DNA, which scientists are discovering, and you haven't even, I heard that apparently the information is just so much information in the DNA that, we can't even comprehend it all. You're still learning.
Yeah.
And it's so orderly, and it's reproducing itself.
Yes.
I mean, where order comes from a creator.
Yeah.
I mean, if I had a book in front of me, did that just happen?
No,
it had
I'm not.
to
I'm not.
have
I'm
an
not
author,
saying I disagree with you.
It had to have a printer. You had to have a binder, and so it all happened like that.
Yeah.
The same with the human being. I mean, I don't even like the question to put a question like, how did it happen? Scientists have got the evidence that it couldn't possibly happen by itself. Random and all that. It had to be created by an almighty God. So there's no question. So I'm quite offended that there should be a question.
I haven't meant to offend you. My goal for this talk was to come at this completely unbiased. That's my goal. I'm not saying I find it necessarily easy. That's my goal. Okay, so don't be. What I'm saying, I'm not trying to actually tell you which way it went. Okay, I'm just delivering what I know and what I've read. Okay.
Yeah, but we're hearing it every day actually, I was coming here tonight to think that you're a young Christian man and that you were going to be used of the Lord to show that creation is proven by the DNA, and it's exactly as God said. He created each kind according to their kind, and you have the DNA proof. So, I'm not interested in hearing about evolution, because I know that's a lie.
But you need to know what the argument is.
I do. I've listened to it a lot of times.
Okay.
We have a discussion time after the talk.
So I think you should finish.
I think if you need to be able to rebut things, you need to know what you need to be able to rebut, if that makes sense. So I don't think we expect people to listen to us. in terms of our side of the story as Christians.
I can't hear what you're saying. You have to speak louder.
Sorry. I don't think we should put our fingers in our ears and say we're not going to learn what people think and why do they think that. Because I don't think we're doing ourselves any favours doing that. I'm not trying to convince you that God didn't design it all. That's not what I'm trying to convince you tonight. I'm trying to present what people believe or what people think, and I want you to take home how amazing this stuff is, and I want you to make up your own minds. I'm not here to tell you what to take home. Okay. All right. So evolutionary, a guided evolution model is becoming increasingly popular amongst various Christian denominations. So I think the Catholics are embracing it quite a lot nowadays. Yeah, so it's interesting.
Excuse me.
Yep. Thanks for coming. All right. So what other things have people come up with to describe how some of these things might occur? There was a man by the name of... Oh, let's move on, sorry. Now, Carn Smith, he proposed that some sort of non-nucleic acid start must have been an essential part of this if it did happen by itself. Basically he was looking at various clays and what things may have been around back before, back in the early universe, as some people postulate. He was suggesting basically because these crystalline structures, they grow in a particular way where imperfections effectively are maintained, and so then if the crystal is broken and then it continues to build itself as crystals do, that there would be some sort of a replicating system which stored information. So if you could consider the defections within the crystal a source of information, and then they also replicate by being, say, broken in half, and then they would grow again. So there's some sort of a system where you're getting some information that is being preserved and replicated. Okay, so it hasn't carried a lot of weight in terms of origin theory because it's very difficult to prove, and in my opinion, starting off with a non-nucleic acid system, it just seems to not really work, because you can't, it's sort of like making a clay person and then expecting all the clay to turn into cells in reverse type thing. You sort of have to build from the ground up, in my opinion. But if that's how it happens. Okay. Another test that was done, this was in 1952, was to do with seeing if amino acids could be produced in sort of hostile, rough, early environments. As postulated, the Earth was once very hot and stormy and volcanic and whatnot. So they set up an experiment where they had simple gases basically in a test tube, and they were using a spark to try and see what they would get. So they did get some amino acids produced from it. Actually, they got amino acids, but as you could see from the, if I go back, amino acids were the finished product, right? So what they were basically showing was if you've got a chicken and an egg problem where you can't get the chicken without the egg or the egg without the chicken, then if we can introduce the chicken, then it may be able to produce the DNA, that sort of thing. So it's addressing the chicken and the egg problem, but to a very simple end, and you know, so that people that believe that sort of thing, then it gives some sort of it confirms, I guess, that you can make some of the building blocks of life in a test tube, I guess you could say. But I mean, that's not necessarily that hard. There's about 500 amino acids that we know of, and only 20 of them are used for human life. And even then, you've got left-handed ones and right-handed ones, and so yeah.
So all life is made out of left-handed, isn't it?
Yes, I think that's right.
Left-handed.
Yeah.
And so when they actually, but when they did the experiment, they got a mixture.
Mixture, yeah. I think about half and half. Yeah.
And so pretty useless for actually constructing.
Yes. Yeah, that's right. Yeah.
Why is it used? Why is that used?
Because if you mix both together, you don't get. You need all left-handed to actually make it up. And the fact that you produce a mixture means you can't.
Or they interfere with each other.
I think so, yeah.
Because you could get you can get the left-handed ones doing their job, right-handed ones doing their job.
That doesn't work. Doesn't work.
I've heard that they, a bit like carbon monoxide and carbon dioxide, if you're breathing it in, I think they both occupy the space in your red blood cells, and if it's too much carbon monoxide, then you cark it, and if it's the other way, or that sort of thing, I think. Something like that. But I could be mistaken. I think that's something I heard quite a long time ago, so whether that's been...
That's been using that people, I can't think of the name of it now, when they were born, they didn't have arms or things like that.
Not sure.
I understand that had right-handed DNA or something like that.
Oh, okay.
That caused that problem.
Interesting.
It didn't purify well enough in the section.
Right.
Making the pills.
Okay, interesting. Haven't heard of that before. No.
You might never hear it again, but that's
Yeah, well.
But I think that's what, I'm pretty sure that's what he told me.
Yeah. Yeah, so the more tests were done in 2009 trying to go to the egg rather than the chicken, I suppose you could say, and they did similar tests, and they were able to get some very fundamental nucleotide molecules produce, but I think that it wasn't as straightforward as that. I think they had to massage it a lot to get things working. But they're working on that as well to see if that's at all feasible as well.
There's the other problem is that it's generally accepted that it doesn't match the experiments that were done that actually produce some results, don't match the early environment.
Yeah, so because I think it was done, from what I was understanding, it was hydrating and then dehydrating and radiating and so I've said here sunshine, rain and lightning. I thought, did they add excessive pressure or something like that as well? Because I
think it's the level of methane that is reduced.
Okay.
And then you've got radiation and stuff like that.
Yep.
That would have actually killed the early. That's even more recent.
Yeah.
This was still in my son's biology textbooks 10 years ago.
Yeah.
Even though you read everything, they're basically described it pretty soundly.
Yeah.
None of the serious scientists actually believe this one. They go for panspermia or something like that, like Dawkins and co. They don't actually get any credit.
Yeah. Okay. I was reading up on this, and obviously I'm reading slightly out of date stuff, maybe, but I guess my take home point here was that they're trying to work out ways to go as fundamental as possible, because I think only building the end product without having the pathway to get there is probably quite fruitless, if that makes sense. And the other, I'll brief over this one, and then, that's pretty much done. So, the other abiogenous methods that I've looked up are basically RNA world, which fascinated me, I guess, in the fact that if there was RNA originally, however it got there, whatever, this RNA is capable of both having information and self-replicating. And from what I understand, there's still a lot of massaging that has to go on in terms of intervention to get them to do these sorts of things. But that's another theory that evolutionists or old Earth creationists are bringing to the table, and it's interesting. So,
does this RNA replication actually happen now?
Yeah.
Or is it just kind of speculation? Is there
I have seen models of the RNA actually producing and replicating itself. That's right. So whether it's speculation or whether they've actually proven in the lab, I can't tell you, because I'm not sure. From what I've seen, they've even had software out there where you can design your own RNA, and it will fold in a particular way. And they not that long, might have been a little while, a couple of years ago now, but they were actually putting it forth to public people, the general public, to design RNA to see what the bonds would do, how it would twist and whatnot, and to design an RNA. So what they were doing with that, I don't know, and whether anything became of it, but it was sort of a thing a few years ago that people were trying to push. So I didn't get onto it because I think it was taken down by the time I had a quick look at it. But I guess the interesting part about RNA is that it is sort of an intermediate stage between data and practical use in terms of actually doing something. So it has the best of both worlds, I suppose you could say. And other theories here came from space, and some other sort of things along those lines, but that'll do. I had some closing thoughts and questions, which my lovely lady here might have been interested in, but is there a limit to the complexity we see whereby natural means are inefficient to explain them? So in terms of, what I covered was really a very simplified and dumbed down version of what we see in the world around us as we look deep. And my second sort of question, I guess, to pose is, can all the mechanisms described, all these different enzymes and whatnot, be a product of gradual changes? Is that actually possible, or is there cases where any length of intermediate phase, or any length of some sort of intermediate molecule just wouldn't cut it? It just wouldn't do the job that's required. I'm not sure if I'm asking the question properly, but so a very complex chemical which has to be this exact line of amino acids, if there was just one or two amino acids different, it acts completely different. So are those things explainable, I guess I could say, with a gradual change system, which I struggle to get my head around that if the answer is yes, because I can't see that being possible, but that's my take.
That RNA, I think some see that as an intermediate
Yes.
itself in production of DNA.
Yes.
The RNA is simpler, and it's just one of those steps along to more.
So RNA is interesting in that sense.
Mm.
Yeah, that's right. So but RNA didn't go for that long, if it did in that model, in terms of by the time it reached, on a cellular level, it was out. So it's still from those very basic beginnings that it only explains before that. It doesn't explain even after that.
And when you say simpler, it might be simpler, but it's still enormously complicated.
Yes.
Yeah. And so the third point is it at all reasonable to hold on to an intelligent design given the mountains of evidence against it? I've read up and watched plenty of debates and people talking about this stuff online, and the general consensus is, if there's one speck of evidence for any sort of intelligent design, there's a whole mountain of evidence over here for not intelligent design. Is that actually the case? Sorry, I'm reading off my slides and you guys can't see. Oh. Yeah, well done. Well, that's what I'm asking. People say it. People claim it. I mean,
what's one point of this evidence? That line of mountain.
Well, that's what I'm asking, I guess.
There's a few engineers present. That's good. And I think we have a special perspective on life, because engineers build systems, and you build big, complicated systems. We know that I've been involved in big projects, and invariably they go bad. You have to kind of get it all right, else it just doesn't work. And to actually kind of look at a biological system and see how it's all fit together so intricately and how it works. I think engineers have really struggled to produce something like that, because invariably big systems go wrong.
But isn't that is the case in biology too? That's natural selection. That lots of different critters will come along and they'll die out because they're just not fit for survival.
Yeah.
But one might have a slight advantage over the other, tiny advantage. And if that's in the genes, it's then passed on to the next generation. And all of complexity can, in the animal world, in the living world, can be explained that way.
Natural selection doesn't increase complexity, it culls the
That's right.
So it reduces, it doesn't increase.
No, it reduces. The selection doesn't. Replication has the ability to build complexity through change. So you replicate things and you duplicate systems, and that happens through replications of bits of DNA. The whole system is designed for this property of replication. And when you replicate systems, you have the capacity for some systems to be diverted to a different function. And if that's advantageous, you can select for that, and you can select for multiple functions from where you originally had one. That's through operational natural selection combined with replication, and that is how evolution by natural selection is perceived to be able to build complexity. It's not inherently contradictory.
Amen.
To be fair to the theory, like natural selection reduces, but the mutation increases, doesn't it?
But selection enables differences to diverge as well.
But all this is kind of after the fact. That process is after the fact.
It is. So it's a bit like.
But the DNA, protein, and RNA cycle precedes all that.
Oh, it is, and that is a mystery to evolutionary scientists, exactly how the thing got going. There's been an enormous lot of mysteries solved in terms of how it works and how the whole process of natural selection and evolution can work, and that's enormous. The understanding of that is far beyond what it was. And so one of the I think the key points that came up was about a whole swag of evidence against intelligent design from the DNA. And people
Sorry,
say, well,
all right, what's the nature of that
where
evidence?
is this swag of evidence? Well, one of the things that flows on from what has been explained tonight is this redundant information that's in the DNA. So the redundant information is about, I think Joshua said 90% of the DNA is actually not only not coded to make proteins or enzymes, but it's not actually used in any way at all. So some of it, you talked about some of the redundant stuff being used for other functions. Some of it's not used at all.
When you say it's not usable, it means they haven't worked out how it's used.
Yeah, they haven't worked out how it's used, but what they do observe with it is that you can cut bits out, and so there are various things you can either manipulate and do, but there's also natural processes to do the same thing that comes along and takes a chunk of DNA, sticks it in here. It doesn't affect the organism if it's done in these redundant bits. Apparently it doesn't affect them.
Yeah.
So but what you actually observe in these redundant bits that are not actually coding for proteins and things is randomness accumulating. So you get a lot of different random stuff in there, and you can change it, and it doesn't seem to have any effect. And so biologists use that as an evolutionary clock. So if you actually take the huge diversity of organisms we've got and look at features that they share in common, basically the whole nature of life is that, in terms of its similarity to itself, both in terms of just the physical attributes, you can fine-tune and everything else, but it's basically a tree-like structure. It's hierarchical, where you share certain things in common, and the further, a lot of things have a little bit in common, and then down the structure, you get more things shared in common. And that tree structure actually is borne out by these redundant, if you actually look at the redundant bits of DNA that have these properties, you find that further, less related things appear to be, you get accumulation of differences, of randomness. So that's not something, and also mistakes. So you can actually see things that are non-functional. The functional bit here, something stuck here and it's non-functional. These functional mistakes actually appear in this hierarchical structure of living things. They suddenly appear here. And because of the relationships you have, you can actually see, this mistake appeared here, it'll only be in here. Now, if these things were all made in terms of design from the beginning, they will have a similar level of mistakes. You wouldn't be able to use this randomness as a degree of separation of one thing from another. But no, you can actually look at the organisms, you've got an idea of how closely related they are, and when you actually look at the numbers of differences accumulated, the more closely similar they are, more related they are, the less of these random differences and the less of these mistakes that appear. And this not only the non-redundant in the redundant DNA, it's also in the fact that some of these base pairs, some of these amino acids, you can have different combinations of the base pairs to code for them. And so things which are basically very similar will tend to use exactly the same, even though they don't need to. But if you actually look at something that's not so related, lo and behold, you find that the base pairs, there's more differences in the base pairs. They don't have to be there, it's just random. But the further back you go as to how things are related, the more of this noise gets accumulated. Doesn't add up for an intelligent design to say, I'll increase the randomness artificially. No, it fits with the fact that these things have actually developed from a hierarchical relationship. They've actually had a common ancestor. It's this common ancestry which then divides. And it's replicated in the DNA. That's what we observe, that's what scientists, and there are thousands and thousands of scientists around the world who are coming up with these consistent pictures. When you actually look at the DNA, and compare the numbers of differences in base pairs. So you're looking at thousands and thousands of sequences, and you compare them. And this is what scientists do, biologists are doing all around the world in laboratories. They are actually comparing in detail, and it's to such a level that basically they have a time clock.
Excuse me, you're talking about evolution and very broadly DNA, but we have got so many different kinds of animals, trees, and human beings. Each one has their own specific DNA. And have you ever seen a dog change to a cat? Now, you talked about, the survival of the fittest. Now, that's within the kinds. Now, Cats change to different kinds of cats. Dogs change to different kinds of dogs. But human beings remain human beings. There's not animals changing to human beings. There's no evidence in the world where one kind changed to another kind, and that's one set of DNA. It's not just one DNA, it's each kind has got its DNA. And you can't say that that changes. There's no evidence that there has been any change. And that's what we're talking about. It was all created as each kind, and human beings were created as human beings, and apes and monkeys and all that were created as apes and monkeys, and they've got their own DNA, and that doesn't change to another DNA.
But DNA, by its very nature, is designed, if I use the word, that God has designed DNA in such a way that it is very, very good at changing. It's very good at conserving
Not from one
things.
kind to another kind. God created each kind. He says that in His Word, and scientists have shown that when they study the DNA. And even today, the scientists have shown that all races come from the one, from Adam and Eve. They've shown it. You can have black, white, and yellow and all that. They've all come from the human DNA that was in Adam and Eve. And that's been shown scientifically.
Actually, I've no desire to upset you, but that's certainly one of the areas that's being discussed and investigated right now, because that the best knowledge so far, I'm just taking your comment that science says. So science says at the moment that the base population from which we all come must have been around about 100 or 200 pairs. So just that's what science says. I just want to correct you just on.
Well, sorry. So
Yeah,
what do you mean? What do you mean by that
consistent.
statement? You said it must come from 200 pairs. What do you mean by that?
For the diversity that we've actually got, and for the tree, they can track it back. At the moment, the best science, I'm not saying this is infallible, I'm saying that the best science is saying that the population from which Homo sapiens must have been from 200 or 300 breeding pairs.
Where did that come from?
They came from their ancestors.
So that goes
So you're saying
back
there's a
further,
bottleneck.
right? But that's to where we are for Homo sapiens. I'm just commenting on because this is a whole lot of stuff we're going to read about Adam and Eve and stuff in the scientific literature saying it couldn't have been a single couple at that time, right?
The consequences of a population being reduced to two individuals that has on the variation on the level of what's called homozygosity, where you have deleterious genes, you don't get the genetic diversity. So if the human population had crashed down to a level where there was only two individuals, or we'd come from just two individuals, then we wouldn't have the variation that we have.
So
that's what science is actually saying.
I'm just commenting on science. I'm not commenting on the theology.
So it is saying, science does say we have
science, not the
No,
theology.
a single origin, that we had an origin from a single population.
That has to come from the same one.
Mixed a bit with Neanderthal, a slightly different species. But
I was just going to, how did the first 200 kind of appear from which you...
Well, they came from our ancestors. So there was
Turtles all the way down?
Hey?
Turtles all the way down?
It goes all the way back to the beginning of life.
In theory, you go to
Because it's
whatever was the
populations
primate
that carry on the...
Yeah, just say
the
it one at
genes.
a time.
Sorry.
Just let Peter go.
Yeah, it's populations that carry on the genes.
We got
It's a
genes.
very precarious thing to have one individual or two individuals of it. You know, it happens, but it has serious consequences.
According to the biblical record, which you can believe or disbelieve as you choose, in the beginning when God created, He created them as perfect. So therefore, genetically would have had a perfect genome, so to speak, with all of the potential within it for what's come afterwards. So therefore, having just a single couple in that context would not be a problem.
I don't think it says perfect anywhere. I think it says God was pleased. God was
Was
pleased
very
with what
good.
He made, very pleased with what He made. But I don't have a problem with that.
But understand
And that doesn't mean perfect. I mean, I guess one of the things that the the things we see as
Thank you.
mistakes or mutations are actually part of the process. They're part of what God used through an evolutionary process to the system that we have that works, that's, I believe, in the mind of the Creator from the very beginning, is a system which actually works with change. So there's a lot of the capacity. Like, how do we understand this capacity? There are some Christians who would say, we've all come downhill since Adam. But there are also Christians who would say, yes, we accept some microevolution. This microevolution is about this variation. It's about having some randomness and flexibility built into a system instead of designing a perfect organism, which would be fine if you had a perfect organism suited to a perfect environment. There's no need for sex. I mean, the whole issue of sex and replication is to basically be able to have something that copes with change.
Nothing. It's got to do with creating more people.
So this flexibility that we see that even creation, a lot of creationists will accept. Oh, well, God made them so that they could actually be adaptable. Is actually built on this system not running perfectly. But this whole concept of something being perfect and something, no, it's a system that God works through a fallen world. He works through what we have, and what we actually observe is this world through which God does amazingly creative
Again,
things.
biblical perspective, this is the world. Later.
Can I make a comment on where the discussion's gone? We've kind of gone down the track, all right? We've gone down the track and we're talking about evolutionary theory. But all this stuff on DNA replication, doesn't that precede that? Like,
It does, and my whole
how
point...
does the DNA, RNA protein cycle start in the first place?
My whole point is that with the replication of DNA, mistakes get replicated.
Yeah.
And DNA has the capacity to absorb lots of mistakes. From what we learned tonight, it has the capacity to absorb variation, mistakes from what was the original thing. This is what this has. These three letters code for this, but you can actually have some variation in some of those. Mm-hmm. And if a mutation causes one of those to change, you still produce that amino acid. That's fine.
Look, I think I hear what Kevin's trying to say is that that's not what the purpose of this thing is about, because that's already accepting that DNA exists.
Yeah.
So
No.
That was what the step is. What is the step between nothing and creating a single cell that I was trying to...
That's a different question. It's a different question.
But that is the question.
That is the question. We've tried to answer tonight.
The single cell is highly complex, which has to be...
I think given my questions up here, it does expand a little bit on that other end too.
Yeah.
So.
It just tweaked to me that that's what you were trying to say.
How do you get from chemistry to DNA?
Yeah, so...
that's one of the questions, but I think just the amazingness of DNA and how it works, which says there's a big gap there, and we can basically say, well, God must give us a gap, therefore God must be the only one who can fill it. Or we can look at what we've already understood about how it works and say, well, yes, we would have called that a gap that God had to fill in the past, but we now understand amazing things about how the cell works.
Yeah, but in fairness, that is making a presupposition that we've learned this much. So we just figured that bit out as well.
Well, we may not. No, we may not.
That's what I just want to bring out. We may not, because
there's a kind of the God of the gaps, and so Christians are kind of all sorts of people are accused of making God fill the gaps. But then there's a converse of that, and that is we currently don't know, but one day we'll find out.
Yeah.
And how do you know that that's the fact?
That's a valid point.
Because like the God of the gaps can be used to explain anything, and one day we currently don't know, but one day we'll find out. Can explain everything too.
Well, that's fine. But I guess what I'm really saying is that
if it explains everything, then it explains nothing.
What we learned about the structure of DNA tonight answers like the claim was made that there's a whole weight of evidence against intelligent design. What is that? Well, a very powerful bit of evidence because of what we now understand and what we know about the nature of how DNA works. It replicates mistakes. When you just look at where those mistakes are placed in the tree of life, and it's just totally amazing. That is the weight of evidence.
Yes. There's a bit of confusion over the term intelligent design. Yes, right. Intelligent design is also known for a very specific movement that comes out of the Discovery Institute. Yeah. But then it's also used in a broader sense. Ultimately, is there an intelligence behind the design of the universe? Yeah. And that's a different thing.
Yeah. But my point is simply to say that if God was designing organisms and they were basically created at the same time, even major groups of organisms, you would expect them, this bit does this bit does this bit does this. You would expect him to make it so that they were consistent.
Why?
Why?
No, ask the question why.
Why would you? Why would you expect that?
Because that's the way that it works. I mean, why would you build a mistake into something when you didn't need to?
Hang on.
You do things.
Mistake's just your word for it.
No, the mistake is a random thing that's happening because of replication.
Well, it's
not because of replication. It happens because of mutation. It happens because bits of DNA get cut out and stuck here.
Without being
But if you find a bit that's been cut out from here and stuck here, and it actually does something useful.
Yeah, well,
then you say, oh, I'll replicate that. Or if it's been cut out from here and stuck here and it doesn't do anything useful, why would God repeat that across the whole
you never know what's down the track of evolution, perhaps.
Why would God create the universe? So this is the one of the arguments against God that you often hear. Oh, God is so wasteful. Look at the size of the universe he has to create to just create Earth. So it couldn't have been done by God. But that's just to make the assumption that God is an engineer, not an artist.
Yeah, and I think the point that I'm making is not actually understood. This is a bit like God writing a history, making up a history, painting a history, so that all the things were made looked like they actually had a history. So there are some people, for example, who will say, well, the fossils are all there. God planted the fossils there to make it look like there was a past when he created stuff. It's a bit like saying these strange bits in the DNA that don't make sense were put there by God to make it look like there was an ancestry, but there wasn't. And that, why would God do that?
That's not my question. That's
not your question, but that's what I'm saying.
Hang on,
the pattern that's there begs that question. It begs that question because there is a history in the mistakes. If you look in the DNA, if you look at the evidence, and I suggest anyone who's interested in trying to understand what I'm actually saying, have a look, go to the BioLogos, which is a website produced by evolutionary creationists, evangelical Christians who believe in evolution as a process. Go to their website and look at the blogs by Dennis Venema called Letters to the Duchess. He goes into great detail, which I'm really struggling to kind of explain this evidence of these mistakes that fall into a pattern that reads like history.
What you're saying is that...
And God would not paint a history that didn't exist just to mislead us. And yet the evidence is overwhelming.
A mistake goes back multiple generations and on different branches of the line.
That's right, and it's confined to those branches, the things which are related.
You'd also...
And it gets replicated within the branches.
So I'm not disagreeing with you. I'm very familiar with BioLogos, right? And I'm not at all anywhere near as equipped as what you're saying because that's not been my main area of research. But I'm very familiar with theistic evolution, right? But I'm just calling out the presuppositions that you're making as you make them.
That's quite valid.
That was all I was trying to, because I don't know that I necessarily disagree with what you're concluding. I'm just saying that there are some very presuppositions that we want to surface.
Like it's junk DNA, for instance. I mean,
that's another
if you say
like Michael Behe,
an opposable thumb might, not necessarily
the reasons,
but
the reasons, guys,
you say
rail against that.
this mistake's been copied for all these apes and humans, and this is where the line was, but it may be a particular pigment in the eye or something like that, which is because we are very similar structurally to them. It may not actually be a mistake.
What are some of his assumptions that you take issue with?
Well, the first one is that we called out that the science will get us all the way there one day, so we've got a perfect understanding. That was one.
Yeah, and I don't necessarily
the second one is that junk DNA is actually junk DNA. Right, we're finding out less and less. But certainly, I'm familiar with what you're saying about the unused portions of the DNA and how they turn up in the tree of life. But if you listen to somebody like a Fuzz Rana, and I'm not versed enough to actually be able to get into these discussions. But if you listen to a Fuzz Rana directly addressing Dennis Venema, right, and BioLogos at times, or certainly Michael Behe, right. they will argue against some of those assumptions of junk DNA and whether that actually leads to a single tree of life. And these guys are evolutionary scientists, but that's the edge of my expertise. I'm just calling out what I've heard in myths, but calling out the presupposition. I'm not arguing with what you're saying. I'm not arguing with what you're saying. I'm just saying there's a larger context that we want to ask questions in.
Yeah, just to paraphrase what you're saying, just to appreciate that we're not totally deaf. You were saying that the appearance of randomness does not look like design, that there's a kind of a random process underlying it, and there's evidence for that random process.
What he's saying is that if you take a mistake in our DNA, or what's considered a mistake, so there's a big botch up somewhere in the 92% that's not used, and if you take that as one big munted gene or something like that, and that, say, a chimpanzee has that exact same botch up, and then 3 generations of whatever that is back has that same botch up, and then nothing else does. So it's like it happened at that particular point. And then if you take another botch up, it might go back 5 different
all the
lines,
might have
and then it spreads out from another particular point. So that's what he's saying.
So he's saying it doesn't look as though it's directed by an omniscient
That's
design.
a common ancestry then? Are you saying, I'm hearing you're saying it's a common ancestry then?
Yeah.
That's the conclusion that's made. That would be assuming that it is actually a botch up and it's not a particular design.
Both the presence of mistakes and also the accumulated amount of random differences corresponds to that pattern of shared, speculated, but shared ancestry.
So what you're saying is that if we all had, for instance, where the first 2 nucleic acids or pairs were coding for a particular amino acid, and then any of the third one could be anything and it would still give you the same amino acid. You're saying that, say, us and the chimpanzees and maybe another couple of them have typically one of those, and then fish and something else has another one of those, and so it looks very much like that mutation was shared amongst a family of apes.
And if you're looking at a functional gene, things that are really closely related won't differ, but also things for the same functional thing, things which are much more distantly related, they won't differ very much in that functional gene either. You can't afford to have variation in those genes, otherwise those functions won't work. You take a piece of what's believed to be non-functional redundant DNA in 2 very, very closely related things that everyone would agree, probably had a common ancestor, even the creation people would say, and you look at those and there are a small degree of difference. You get some things that are slightly less closely related. There's more differences in the redundant DNA. You go back further and you find some of it's all the same, some of it's all the same, but the total amount of accumulated differences is enormous if you go further back, because that can afford to accumulate those random differences. And that's the way the DNA is actually telling a story.
Can I ask a question? There's 2 things that I don't even read about this, so I'm learning a lot, but there's 2 fundamental questions for me. Is if we intermarry in our families, then there's problems. Okay, so that hasn't been explained. So that says to me that there maybe wasn't just an Adam and Eve. Okay, because if there was only the 2 of them, then somehow the incest problem.
Well, that's exactly the population getting to.
I'm not asking. That's just a question that I don't think has been addressed. I've lost the. Sorry, I've lost the other one. It's more about. I guess it's that there was. So, what was the common ancestor of apes, and what was there common ancestors before that? Was it a tree, with a single stump, or was it parallel strands? Is that what they're saying?
That's what the evolutionary scientists believe. It's a diverse population. There's always, you don't get. So
we're related to the plants somehow.
What they say is eukaryotic cells. That's where it divides, and then there's the prokaryotic cells. There's a lot of stages in it.
So Peter, can I ask you a question?
Caveman, say, can you produce, a 2012 Mercedes Benz from scratch? And you leave the caveman there, at that point in time. No, there's no way, no matter how long you leave the brightest caveman there, they're never going to come up with an automobile, that functions the way a modern car functions. You basically, and yet, the way it got there was through a whole lot of, and you can't visualise. You look at this car and you think, how could that ever be put together? Because this won't work without that, and the thought that's gone into this is amazing. You can't just do that by gradual processes. Caveman's not going to get anywhere even there by gradual processes. It doesn't matter how long you give him, gradual process won't get there.
You're suggesting the city's
it's actually a whole series of steps that you can't visualise looking backwards, whereby something, steam power was invented and then, rubber was discovered on, there's a whole network of things that you would not guess by looking back. But there is a pathway that links them all up to come up with this car. And that's, and we may not, if you start with a modern car, look back, how did this actually develop? We might be able to go a long way understanding a lot of how it works and how it comes. You may not get that initial first step or whatever that came up with the internal combustion engine. I don't know. How it actually, humans, made iron, metal for the first time, all this sort of stuff. It's a bit like that, looking back saying, well, we can't see how it works.
I guess it's if the car was then needed to make, I guess there's still the chicken and the egg problem rather than just a linear progression. It is a chicken and an egg problem.
Now, can I just ask you, because given the position you're taking, I'm not saying it's wrong or anything, how do you integrate that with your Christian faith? Because it's not, because not sufficient to knock you off your perch, is it?
What I'm trying to do is redress the balance. I work as a biologist. For a long time when I was younger, I struggled with reconciling it with the creation account in the Bible. And I felt at that time that God made it clear to me that I should not take this as an obsession. This wasn't to be something that I became preoccupied with. So hopefully, even though I've been pushing that particular view, I'm trying to explain the science view of the world in which I operate and work with. It makes a lot of sense to me. I think the only way that I can reconcile it with the biblical account is to say this is not a scientific account. This is a story that represents truths. God wasn't trying to teach us how He did it. I believe in miracles. I think science. The science is not there to kind of prove miracles. It's not what science is about. As much as science can understand a process, and I think some of the understanding that we have through this evolutionary process is quite amazing. But if it ever came to a point where it said, well, this means God doesn't exist, I would say, well, that's nonsense. I don't believe that.
It's a long way from that, though, isn't it? Really.
It's different. It's quite different. It's something quite different.
It is. You're right.
Well,
that's what the debate.
Why did God do it this way?
There are a lot of people colleagues, whatever. I think it is important. So even though God sort of laid it on my heart, you must not get carried away with trying to convince people one way or the other. And I hope I'm not fall into that trap. I think one of the concerns that comes out in Biologos is there's a lot of people who will use very simple Christian beliefs, particularly biologists that I work with. I reckon biologists got one of the lowest rates of Christian profession. People professing to be Christians of any
Geologists are lower.
Is it lower, really? Okay.
I reckon it is.
And I think
that, but this
it would be a real pity if the story in the Bible is actually not really about telling us how things happened or whatever, and people are basically dismissing God because of that. I guess
why do you think there would be a creation myth or story in the Bible at all if it's not the intent? Is that because people thought that they should put it in there just to make it nice and well rounded?
It's very important to see God as creator. I think our relationship to God starts with God as actually being the source of everything that there is, who is outside of us, there's no question. If God is creator, that says a whole lot of things about our relationship to God. We are answerable to someone who actually had the power, the brilliance to even think up a universe where these amazing things, and you can look back and say, maybe this happened like this. It all makes sense. Evolution at this level, this far, makes sense. I can't go all the way back to the first appearance of life, when you think about self-replicating molecules that somehow capture themselves and get, a system that somehow works. But regardless, it had to be in the mind of God to come up with something that works. The fact that anything exists, God as creator is such a fundamental thing, and it talks about our relationship to the person that's made us, and the whole moral issue then comes into play. And this story in the beginning, when people say there are two stories there about the creation, the first creation account and the second creation account, and you can interpret it that way. I'm not saying you can't, it's all open to interpretation whether you take it literally or not. The whole moral dimension has to come out. So I don't have a problem with it.
The biologist folks would say that the creation story Genesis is an ancient account.
Is a what, Kate?
Ancient
account.
Yeah, Christians would say the same thing.
An ancient account.
It is an ancient
That's
What does that mean?
it's take Professor John Lennox's example. He said, you could describe an event where you've got an energy transfer in a steel plate into a confined area, and then after enough heat transfer has happened, some molecules escape, and you actually get a whistling sound because of jiggling of air molecules that hit your ears, right? So the agent account of that same thing is, well, my wife put on the kettle because
You said agent account.
Agent account.
I thought you said ancient.
Yeah, I thought
agent account. So one describes it, they're describing the very same event, right? But one is actually describing it from one perspective, and the other is describing it from an agent.
Yeah, I wanted a cup of tea.
Yeah. It's like John Lennox also asks, how do you explain the jet engine? Is it Sir Frank Whittle? Or does Sir Frank Whittle compete as an explanation of the jet engine with the forces and description of physics behind the jet engine? No, it's a different description. I don't know if that's. I think that's what I've read a little bit in Biologos. And does that mean your?
Yeah, it does. I mean, it's talking. I mean, there is some what I would think. I mean, people don't interpret it as necessarily symbolic, but for me, when I read about, the cherubim guarding access to the tree of life, and then encountering the tree of life in John's picture in Revelation. As the tree growing on either side of the river of life, throwing out from the holy city, with trees, leaves for the healing, 12 Asian season for the healing of the nations. To me, that is not a physical tree, and for a lot of Christians, they would have trouble accepting that as a physical. I'm not
talking about meaning, isn't it?
You can't. I'm not saying you can't interpret it as a physical tree, but to me, it makes more sense as a picture. Like a lot of the stuff in Revelation makes sense as symbolic pictures, and yet this same, the same phrase is there as this tree being guarded by a flaming cherubim or whatever in the Garden of Eden, lest Adam and Eve partake of that tree and get eternal life. And to me, that's a picture story. And so much of that account I would interpret as a picture story about what God's on about. And I think God has worked through myths and stories and things, and I'm not saying, I believe in God can intervene in the world in the most amazing ways, and we can't necessarily work that out, and there is a lot that we do need to take from faith, but for me, taking a literal interpretation of Genesis is not something that I feel I must take.
Where do you think biblical history begins?
That's a very good question. There are difficulties in drawing that line.
We actually had a talk on that.
Yeah, Tim Patrick.
It was after Noah, I think.
Like when is it? If the beginning is mythical, at what point is there the transition into history?
You can actually relate it
and
to
looking backwards,
other accounts, or
it's a problem.
He actually did quite clearly define it. There's people that think that through, and they define that change, don't they?
He said he had difficulty defining that.
Exactly where it was, but
who did?
It's sort of like
Tim Patrick, yeah.
Where because to me, I had trouble with Noah, for example, because that's like it's starting again. Our real ancestor is Noah rather than Eve. Really, if you wanted to take it literally. So that is, and I think he was hinting at somewhere around there, and he was saying it's where historical, where you've got things like judges and stuff like that, where it actually relates to things
real
that are going
events.
on. Yeah, where
documented events.
That's right. Yeah. So it's a bit vague around the Ten Commandments and all like that. So that's I think that's what I remember from.
Yeah. We've gone over time. It's all your fault.
You've generated a lot of discussion.
Yeah. I think a lot of the kind of latter discussion was that I was very interested in your last comments about how it fits in with your vote.